Thank you all for coming to hear my talk on open box science today. Let me know if you have any issues hearing or seeing anything. Okay. I also want to give a little disclaimer. I've got a 4-month-old who's decided to just wake up right before this began. So if you hear a little purring in the background or a wail every now and then, you'll know what that is. It's not a cat, it's my child. Yeah, so as Jerry mentioned, my name is Alexa Blanchard. I had the pleasure of doing my PhD in the lab of Margaret McCarthy, who is just absolute incredible woman and a trailblazer in science. I was honored to have spent 4 years working with her, and of the many trainees that she has in her lab are still continuing a lot of the work today. So I'm going to show some published work as well as some unpublished work. So let me just minimize my screen, get this ready here. So I've always been really interested in the role of immune cells regulating brain, brain homeostasis, and in Peg's lab, particularly brain development. And the peripheral immune system has been known to be a player in multiple neurodevelopmental processes, but more famously, they're known for how harmful they can be to the neurodevelopmental processes. I'm trying to click. There we go. They are the immune system can respond to environmental and infectious exposures, particularly in utero, that can cause maternal immune activation, circulation of cytokines, and just inflammatory molecules that can lead to perinatal inflammation. And perinatal inflammation is a known link to the development for neurodevelopmental disorders, including autism spectrum disorder, ADHD, schizophrenia, etc. But more recently, in about the past 10 years or so, what's been really interesting is a number of groups that have come out with data showing that the immune system, particularly the peripheral immune system, is important for physiologic brain development, not just pathophysiological processes. So B cells have been known to be involved with oligodendrocyte proliferation and myelination in the developing brain. Helper T cells have been involved with microglial maturation. And from our lab, we showed that actually mast cells, a very small population of innate inflammatory immune cells, actually played a role in driving sexual differentiation in a part of the hypothalamus called the preoptic area. And I'll touch on that more later. So this was my launching point for my work, was really understanding, okay, you have this very inflammatory immune cell. How is this getting in the brain, and sort of how is it regulated in that space? So I'm sure for anyone here who's heard of a mast cell, the main context you've heard it in is in the context of allergy, where mast cells are known to have IgE loaded onto their surfaces. And when an allergen binds, it causes a massive and rapid degranulation event that releases very pro-inflammatory cytokines and molecules, including histamine, that cause our known and hated allergic reaction. What's not mentioned here is anaphylaxis. Mast cells are also can cause enough inflammation to cause anaphylaxis, which is actually something I suffer from a peanut allergy. So I've experienced the mast cell firsthand. However, what's really interesting about these cells is that they are not just involved in allergy. I like to think of them more like the master cell. They are sort of like a neuroendovascular immune modulator, because they really have the ability to traverse all of these different regulatory systems and multiple different organ systems as well. Through the constellation of surface receptors that allow them to respond to neural signals. I'm sorry, you can't see my pointer. Hold on. Here we go. That allow them to respond to neural signals as well as immune signals and endocrine signals. And in turn, they release a variety of both preformed compounds that are packaged in these granules, immediately accessible to the extracellular environment upon degranulation, including histamine and serotonin and enzymes, as well as a wide variety of different compounds that they can secrete over time, which allow them to have finite control over the microenvironment that they reside in. So I want you to think of them outside of allergy and more like they can be this master regulator across multiple systems. And they're really well known to be involved in different physiologic processes, including angiogenesis, tissue remodeling, immune signaling, endocrine signaling. So they really are just this really incredibly diverse and very interesting cell type to study. They're also found in multiple organ systems across the body. And depending on the organ system and also what tissue they are in that organ system, whether it's a mucosal tissue or it's a connective tissue, they have a variety of different both receptors and also intracellular contents that give them a massively broad phenotypic variability. What's little known, unfortunately, as most things in the immune system, little is known about what mast cells do in the brain. Most of the work has been focused on the adult, where we know that there's mast cells that reside in the meningeal space, which is the border, the tissues out lining the outside of the brain, and in perivascular spaces where they help regulate blood-brain barrier integrity. There's been a few papers about showing their involvement within actual parenchymal regions like the thalamus, but this is really not well studied, particularly in brain development. So when I started off in Dr. McCarthy's lab, I started focusing on this cell type in the preoptic area, where we saw maybe a dozen mast cells that arrive over in the postnatal period and are responsible for sexual differentiation. But when I sort of was panning across the entire brain, I was noticing that there was, no mast cells in the cerebellum, which is this organ in the back of the brain. You have a few mast cells lining the olfactory bulb, which is sort of the interface to the airway, which makes sense that you would have some mast cells there. And then very interestingly, this seemingly large population lining these ventricles and all the way diving deep in proximity to the hippocampus. So if you then flip these sections coronally, you can see these are in 1-week-old pups, both in the ventral hippocampus, which is sort of up here, this bird that's lying on top of these cells. And then all the way dorsally here, you can see that there's mast cells lining that entire breadth of the hippocampus. And they're really residing in this space that I like to call the choroid fissure, which is this lining of the ventricular system that extends, as you can see here, rostrally all the way to caudally, and that these mast cells are really found both in that ventricular space as well as in the lining of the ventricular space. So I refer to these as the choroid fissure or the lateral ventricles, but you really are not seeing those openings right now because histologically they have been compressed during tissue processing, but typically these spaces are wide open. And if you also look back early enough, these mast cells seem to be arriving as early as embryonic day 15, which is equivalent to the 3rd trimester in humans. And you can see here just a few very, very noticeably granulated mast cells in this sort of septal region in the developing brain. And then if you keep tracking that, the same section, postnatal day 1, you see that there's this large population of mast cells here. Day 7, as I showed you before, there's this large population lining here. You can't really see it too well because of the contrast, but they're there. And then by day 16, which is about 2 weeks after birth, you see there's some mast cells here, not as many. And more importantly, by the time you get to the adult, they are completely absent, which really suggests that there's this narrow developmental window here where mast cells enter in the late embryonic stages. They peak at PN7, 7 days after birth, and then they seem to just completely disappear between 14 and 21 days of life. So then not only do they comprise a really large proportion of the immune compartment, almost 50%, which is very unusual for mast cells. They're a pretty rare population. And then they also are numbered in the thousands, but you can see here they completely disappear at that time. So it suggests that there's this tight regulation of these cells entering and exiting this space. And this was work done by Anna Maximova, who is on this call. She's a phenomenal MD PhD student in Peg's lab. She has did some cubic clearing of the developing brain, so you can really see the mast cells here lining. These are actually in the olfactory bulb is up here, the cerebellum is back here, and you can see these mast cells particularly in the meningeal space. But when she goes in and I can't keep my pointer here anymore. Here, when she goes in and kind of deletes all the other parts of the brain and really focuses on the hippocampus, you can see just the density of this population lining this hippocampus, and how many cells you can really appreciate in that space. And just a really beautiful image to represent this population. And the population that is here is what we call piecemeal degranulating. So instead of allergy, where you would expect these cells to be blasted wide open, or in a very quiescent sort of sentinel cell, you might expect them to be fully granulated and just resting. Instead, you're seeing what we call graded as 2s, 3s, and 4s when we're kind of scaling their degranulation. They're sort of spitting off little granules. Like this guy here is a great example. He's just beep, beep, beep. So granules are kind of going off into this extracellular space, but the cell remains pretty fully granulated. And this is a well-documented way that mast cells can exert homeostatic control. Is not through blasting open widely, but it's sort of just having much more regulated and finite control. So both the time period that mast cells are in this peri-hippocampal space and the time that they're active in this space through piecemeal degranulation, happens to be the exact same period of time that the hippocampus is well known and documented to be undergoing its very late embryonic and early postnatal development, where there's this large burst of neurogenesis and synaptogenesis, followed by sort of these connections coming online by day 14. And then the hippocampus is seemed undergoes like pruning after that, but is really fully kind of developed around that stage. And this happens to be when mast cells are here, which led us to this theory that maybe mast cells are participating in hippocampal development, happening also during this narrow period of time. So just to review sort of how mast cells even get into spaces in the body, they begin proliferation in the bone marrow as a progenitor or precursor cell. They exit, one cell exits into the bloodstream as what we call a mast cell progenitor. And then that progenitor then extravasates into tissues where it ends up fully maturing, degranulating, regranulating, degranulating, and just exerting its control. But the important thing to remember here is that typically it's a 1-to-1 to 1 pathway. One from the bone marrow is the same one that ends up typically in the tissue ultimately. So we ask this question, are these mast cells really gaining entry into this really precious, vulnerable developmental brain space through the bloodstream? And so we address this question by administering an alpha-4 integrin inhibitor. It's called CD49D. We administer this to animals every single day, the first week of life, which should be preventing mast cell entry, as well as numerous other immune cells, their entry into peripheral tissues. And we wanted to see if this affected the number of mast cells. And you can see here that although the number of other myeloid, which in our developmental rat model, these are macrophages, the number of macrophages goes down, you can see here that the number of mast cells does not change. What suggests here that, I knew that was going to happen at the same time. Mast cells are actually not recruited from the blood. And then we had to address another theory that was brought on by these series of papers that were published in Science in 2021, which showed that immune cells can actually be derived from the skull bone marrow, drop down into the meninges, which is the outer layer of the brain, and then can actually traffic from the meninges directly through these bone marrow channels straight into the brain. And so we had to ask, okay, well, maybe these mast cells aren't coming from the blood, but is the skull providing them these mast cells? And so we did this by injecting a FITC isomer, just this green color essentially, under the skin above the calvarial or the skull bone marrow. And we're going to track these FITC labeled mast cells that were derived from the skull bone marrow and to see if any of the ones in the hippocampus eventually became FITC positive. And you can see here just 3 hours after that injection, you can see that there's a mast cell labeled here in the skull bone marrow, and you can see all the green in the skull here and even into the meninges here. However, the important thing here is that although the skull bone marrow mast cells become very, very FITC positive, about 80% of them by day 3, you are not seeing these peri-hippocampal mast cells. Pointer again, you're not seeing these peri-hippocampal mast cells at any point really turn FITC positive, which suggests that they are not coming from the skull bone marrow either. Which then led us to this theory, could mast cells actually be proliferating in tissue? Which again, going back to that first diagram I showed you, is really not a behavior typical for mast cells at all. And what we saw when we injected BRDU, which is a thymidine analog to measure the proliferation rate of these cells, you can see here that lining this choroid fissure, zooming in, you see these pockets of mast cells that seem to be BRDU positive. And when you quantify that over time, they're actually highly proliferative. About 15% of them are proliferating. And then that number begins to dip off closer to the time point, that 14-day time point when they start exiting that space. But you compare that in the same exact animals, compare that to the skin mast cells and to the preoptic area mast cells, which are those other brain resident population that we know is involved in sexual differentiation. And it's really astonishing here to see that the skin mast cells are really not proliferative at all. And the preoptic area mast cells are even less proliferative than the skin. And I just wanted to zoom into these two pictures because these were really early on in my PhD. This is one of my very first findings that just kind of blew my socks off and made me think that this is a very special population of cells that you can actually see them dividing in anaphase in the actual brain. And to kind of really solidify this finding, we narrowed this time point in which we injected this proliferative marker to 6 hours. And we actually did this to 3 hours as well. So 6 hours before taking the animals and 3 hours before taking the animals. And then we ran flow cytometry to just see how proliferative this population was, especially compared to the bone marrow, which is again, the prototypically proliferative mast cell population. And you can see that the bone marrow is very highly proliferative, up to about 30%. The skin is incredibly quiescent, almost no replication, and the hippocampus has this little pocket of proliferative cells, which really is stronger evidence that these mast cells are proliferating in situ. They're not having entered the brain as proliferative. They are actually proliferating in the brain, which suggested to us that these might be like a self-sustaining population, which has really not been reported, particularly up to this amount, this very, very proliferative amount, and might be responsible for that increase in number we see around PN7, and then maybe they stop proliferating, which is how we get that decrease at PN14 or so. And here's more work shown by the incredible soon-to-be Dr. Maximova. She was curious, what are the actual growth factors that might be supporting this proliferation? What is allowing these cells to operate in this space differently than others? So we looked at the two common growth factors that mast cells respond to is SCF, which is a stem cell factor, and IL-3. And you can see this SCF protein is higher at postnatal day 0 when we see those high levels of proliferation, but it's also higher in the hippocampus compared to what it is in the preoptic area, which had us thinking maybe SCF was this regulator of proliferation. High SCF levels were allowing these mast cells to proliferate highly. But when we put that to the test, and she injected SCF, IL-3, both, or just saline on PN0, and then she looked at the proliferation rate of these mast cells in the hippocampus at PN2, you can actually see that mast cells did not respond at all to SCF. They only responded really strongly, actually, to IL-3 or to a combination of both. But really, IL-3 seemed to be that factor. So even though it's not more abundant in the developing hippocampus, maybe these mast cells are more responsive to the IL-3 that is there, and that could be really driving that proliferative behavior. And then to further evidence this, she injected SCF or IL-3 at day 0, and then tracked mast cell number at PN7 and PN14. And she saw this huge increase in mast cell number at day 7 that actually persisted all the way through PN14. So it really seems that these mast cells are very IL-3 responsive, and that IL-3 might be that regulator that's allowing them to proliferate in that space. So then we use this sort of to our advantage. Anna then took IL-3 and saline and injected it at PN0, and this is preliminary data that she has just come up with recently. It's really exciting that when you inject IL-3 to try to augment that mast cell number, what she did was she looked at TBR2, which is a marker of proliferative neurons in the dendrite gyrus, which is the hippocampus's prototypic, like, proliferative new neurons are born here area. And she looked at the proliferative behavior of those cells, labeling with PCNA, which is a proliferative cell nuclear antigen. And she did this outline of just the dendrite gyrus. And if you're looking just in the dendrite gyrus, it looks that IL-3 seemed to stimulate neurogenesis in that area, possibly secondary to that increase in mast cell number. And then to further evidence that, Andrew Alley, who's another PhD student in the lab, helped with this experiment where they injected compound 4880, which I'll get into a little bit later, but it is a compound that activates mast cells. So it seems that when you inject the mast cell activating compound, you actually also are able to augment the proliferation in that dendrite gyrus, really suggesting both of these are preliminary data, but they are very exciting and suggest that not only increasing mast cell number, but also activating these mast cells directly might be leading to increased neurogenesis, which might alludes to a role really between that mast cells might be playing in stimulating neurogenesis in the developing hippocampus, which sort of makes sense based off of that timeline I showed you at the beginning of these two events really happening simultaneously. It's very exciting, and we'll stay tuned for more. But then I wanted to ask, okay, so how are these, maybe who knows what their physiologic involvement could be? It could be neurogenesis, it could be many other things that we're all currently exploring. But how are these mast cells different than other mast cells in the developing rat? And so what we did was we did bulk RNA sequencing of bone marrow mast cells, skin mast cells, hippocampal mast cells, and hypothalamic mast cells, and just look to see what the differentially expressed genes are. And what you can see right off the bat is that there's heterogeneity between all 4 of these populations, even the brain resident populations that are very, both in the brain at the exact same time in the same animal are different. And in fact, they are more different than they are similar, which is really, really surprising to us. We expected these to be a very similar population, and indeed they are not. And if you compare the hippocampal mast cells to these sort of canonical skin mast cells, you see this in the hippocampal mast cells, this large upregulation of differentially expressed genes, including CSF2, which is well-known marker for microglial maturation and for myeloid cell activation. MSR1, macrophage scavenger receptor protein that one of our postdocs, Matt, is actually looking into for its potential role in oligodendrocyte development and regulation, and insulin growth factor binding protein 1 and 2, which have both implicated in neurogenesis, as well as a CYP26 alpha here. That's a retinoic acid receptor that we know is important in neurodevelopment as well, and a prostaglandin synthase. So compare that to then the skin mast cells here, where it seems that what is more differentially upregulated in those are inflammatory markers, IL-15, integrin beta-4, CCL1, a chemokine. So it really shows that these hippocampal mast cells might actually be sort of programmed genetically to be more involved with neurodevelopmental processes versus inflammatory processes like you would see in skin mast cells. And then we did a very complex analysis, a network analysis of this data. And I'm not really going to go into much detail here because it's very complex. But I will say that when you sort of look at the programs that are seem to be upregulated in our population, one of them is this module of sort of interconnected genes, which seem to be involved in DNA replication, mitosis, cell cycle, and really ultimately proliferation, which supports the fact that we know that these 2-day-old animals have very proliferative peri-hippocampal mast cells, and the bone marrow also upregulates this module. And then there's plenty of other modules, including module 4 and module 7 from this network analysis that we were able to pull out here. Again, details not exactly important for this talk, just knowing that they're very upregulated in this peri-hippocampal population, and that these modules seem to be involved in cell communication, cell signaling, RNA synthesis, RNA metabolism, biosynthesis, vesicle transport, suggesting that these are incredibly metabolically active cells, and that they might be creating factors very rapidly to be able to secrete to their microenvironment to exert that physiologic control, especially compared to skin, which is sort of that canonical mast cell. So then we sort of, while we're exploring the physiologic role of mast cells and really using that bulk RNA sequencing data set to start driving our physiological functional studies, we also, in parallel, we're also wondering what is the potential impact of activating this mast cell population. And so we did this using what's called compound 4880, is a mast cell activating compound actually that I mentioned earlier. And we are using that compound to mimic a variety of different mast cell activators, such as allergy, prenatal stress, infection, things that you might imagine in a developing pup, either prenatally or postnatally, might instigate an inflammatory response. And if those mast cells, the thousands of mast cells are there, start degranulating and becoming active, what are the downstream impacts of that? And could those eventually be linked to increased neuroinflammation, and thus could potentially be linked to the development of neurodevelopmental disorders? So we explored this with this, just showing you here, this compound 4880 is a very effective mast cell degranulating compound. It works through a non-IgE mediated mechanism, so it's not mimicking allergy per se, it's just mimicking activation, because it causes this massive degranulation. You can see here at 45 minutes, the number of these cells that are 5s, right, that are just fully blasted open. And that interestingly, 6 hours later, those cells look like they begin to regranulate and really form those, their membranes again, just 6 hours later. So it's one single hit, a transient hit that causes a large mast cell degranulation event. And we're sort of looking at now all these downstream effects after that. So the first thing we looked at, because there's well-established relationships in our lab between mast cells and microglia, they're also both immune cells, right? Microglia are the resident immune cells in the brain, and mast cells are these peripheral immune cells that are positioned just adjacent to this developing microglial population. Sorry, my cursor stopped working. There we are. So you can see here these microglia here, and we measured their proliferation rates using this proliferative marker again. And we can see here that administering this compound 4880, this mast cell activating compound, we actually diminish the proliferative capacity of microglia in this space where they are adjacent to the mast cells. And interestingly, we saw a reflexive increase in the number of IBA1 positive cells. And IBA1 is not just a marker of microglia, it's also a marker of all myeloid cells. This includes macrophages and monocytes. I'll come back to that, but just kind of keep that in mind, that this degranulation dampens proliferation. It does not seem to have any impact on morphology, since there was no difference in microglial morphology in the hippocampus after this compound it was administered, but it does seem to kind of increase this number of IBA1 positive cells. So what we then did was we then turned to the most well-known relationship that mast cells have, and they're most famous for, is between mast cells and vasculature. And we said, okay, well, if these cells are degranulating, could they be having an impact on vasculature? And so what we did is just showing here is a confocal image of, you can see 3 mast cells here and all of their granules that are sort of tucked inside the cell. And you see how close in proximity they are to, and just how tightly associated they are with vasculature in this choroid fissure space. So naturally, we had to ask, okay, well, does degranulating these mast cells, do these cause vascular permeability? And to do this, we used MRI imaging. So we injected 4880 in these pups, and we then gave them a gadolinium, which is a marker that's used in MRI. And we put the pups in the MRI machine, and we imaged their entire brain to see if there was any vascular leakage. And to no one's surprise, if you just look at just that hippocampal region, there's an increase in gadolinium contrast enhancement across both medially and laterally across the hippocampus, across all the slices we imaged. And there's increased volume of enhancement as well as intensity, of that enhancement. There's just more enhancement in general in the treated group, both at 45 minutes and 3 hours after. So mast cells do seem to be causing vascular leakage and a compromise of the blood-brain barrier. We then turned to look at chemokines. So we thought that mast cells might be causing release of chemokines downstream. And so the way we measure this was actually pulling CSF from these pups. And again, the important thing to remember here is CSF is all over the brain. The CSF is the fluid that bathes the entire developing brain and the adult brain for that matter. And so when we just look at that CSF in treated animals, we can see that there's this huge increase in chemokines, including CCL2, CXCL1, and CXCL2 that's bathing the entire brain. And also across the entire brain, across multiple different brain regions, you're having an increase in endothelial cell adhesion molecule expression. These adhesion molecules are needed for immune cells to be able to extravasate out of vasculature into brain tissues. And so there's that increase of expression also across the entire brain. Again, this is just hitting this mast cell population in that peri-hippocampal region is having these sort of widespread effects. And then naturally, the next question we had to ask was, is this actually causing inflammation? And 3 hours after injecting that mast cell activating compound, you are seeing this large increase in just the overall number of CD45 positive cells. So overall, just general immune cells. And that number continues to build up to 40 hours, about 2 days after the initial injection. You're seeing an increase in immune cells. And what's really important here is that if you give this mast cell activating compound to a 20-day-old animal where we know there are very little to no mast cells in that space, we are not able to replicate that inflammation. So it really, really suggests that this inflammation is due to mast cells and not due to some off-target effect of this compound. And when we further dived into what cell type is the cell type that's driving that inflammation, you can just see by eye here that the population that stood out most was these classical monocytes, which are the first responder innate immune cells that can then eventually differentiate into macrophages or dendritic cells. But they're sort of these small little round cells that can migrate very quickly to sites of injury or inflammation and can further promote and augment that inflammation. And you can see here that their numbers are increased in the hippocampus at 3 hours, again, peaking at 40 hours and are resolved by about 90 hours. And also what's interesting here is that inflammation of monocytes, the influx of monocytes is not just seen in the hippocampus where those mast cells actually are. It's also seen in the hypothalamus, and it's also seen in that cerebellum where we know that there's no mast cells residing there really. So it's it's like a widespread inflammatory event in response to one single activation event of those peri-hippocampal mast cells, which really just shows the power of these mast cells to induce widespread brain inflammation if they are activated during this time period. And then to really sort of nail in the coffin that this is could be a CCR2 mediated mechanism that's driving classical monocytes, we inhibited CCR2 with a compound. This was done by one of the grad students in the lab who helped out Callie. And she showed here that if you administer a CCR2 inhibitor, that you're actually able to completely negate that monocyte influx. So that these classical monocytes are CCR2 dependent for their entry into the developing brain, which makes sense because CCL2 was one of those chemokines we saw that was upregulated. So we still don't know if this CCL2 is directly coming from mast cells or if mast cells are triggering other cells to release CCL2. But either way, there is a large increase in CCL2, which is then leading to monocyte recruitment, which is really, they're not cells that you want across the developing brain, let me tell you. And so there's a lot more to study here, and many members of our lab are working on this, looking at the sort of the pathophysiological impact of this inflammation downstream. If you have a mast cell activation event that then causes inflammation, well, what does that inflammation cause? Does it disrupt neural development? Does it disrupt microglial development? And we have a lot of exciting work kind of pushing the frontier in that direction. So sort of just in summary, my work in this lab and the projects that are ongoing in this lab really have two arms. One is understanding what the physiologic role of these really unique and very brain region specific mast cells, what they are doing in the hippocampus and in other areas too. Are they impacting neurogenesis, which we have seen that there might be this link between mast cells and neurogenesis in the hippocampus? Are mast cells contributing to microglial development, blood vessel development? There's even some preliminary data in the lab that might suggest a connection between mast cells and oligodendrocyte development. So keep on the lookout for that. And then this pathological role, as I was just alluding to, that there's, the inflammation that is generated from a mast cell activating event really might have these downstream effects on neurodevelopment that could be linked to neurodevelopmental disorders. And as a future clinician, I have to say here, this is where the clinical relevance comes in here, is that in patients that have mastocytosis, which is a mast cell activation and a disorder that has both increased number and activity of mast cells, the prevalence of autism spectrum disorder, one of these neurodevelopmental disorders, is 10 times higher in kids that have mastocytosis. And conversely, kids that have autism spectrum disorder and ADHD have an increased prevalence of allergic conditions. So there might be this link here between mast cells and the development of these disorders. In addition, mast cell activation syndrome, if anyone has heard of it, is a sort of like a newer diagnosis of not increased number of mast cells, but they're very hyper-responsive mast cells. Those patients experience cognitive and neuropsychiatric symptoms, again, maybe alluding to the relationship between mast cells and brain function in children. And there's a variety of different ways that these mast cells could be having this impact, but we're just really excited to have had the collaboration across the lab to be able to both explore these mast cells and to then kind of figure out what they might be doing long-term, both in the physiological space as well as pathophysiologically. And so there's just so many people to thank for this work here. Again, Peg is just an absolute powerhouse, and I'm really grateful to her. I've had many mentors in the lab that have been phenomenal, as well as many mentees that have now gone on, and including Anna, who have now gone on to show and represent many of this sort of preliminary data today. And we're really excited for what's coming out with her and many others. So just thank you across the board to everyone, and I'm happy to take any questions.
Thank you so much.